Hey guys, my name is Max and welcome to Simple Biology. In this video, we're going to talk about bulk transport. Now, in some of our other videos, we've talked about various types of transport, but so far, all of those have involved transport across a membrane. The interesting thing about bulk transport is that nothing actually crosses a membrane. This is very important for certain situations that a cell is going to get into, and we're going to be talking about those in this video. Now, first off, why would cells use bulk transport as opposed to another form of transport? Essentially what happens in bulk transport is that materials are transported using vesicles. Now, if you remember, vesicles are these bubble-like sacs of membrane that are inside the cell. And the interesting thing is that they're completely separate from the rest of the cell. So sometimes that's the reason that cells would use it. If they're going to take in some sort of substance, but they don't want it to just scatter throughout the cell. In addition, a lot of large proteins and other materials are completely unable to cross the membrane. So if you remember, to cross the membrane, it has to be small enough to fit through the transport proteins or small enough to get through the membrane itself. And a lot of large materials, such as proteins, can't do this. And so that's where it would have to use bulk transport, some form of bulk transport, where it would get the same effect, but it wouldn't have to actually cross through the membrane. In addition, there are certain situations where bulk transport gives some greater control over what's coming into the cell, and as opposed to just releasing it through some sort of transport protein. Now, the first type of bulk transport we're going to talk about is exocytosis. Now, if you know, exo means out of or just out. So exocytosis, cytos, cell, so out of the cell. This is the type of bulk transport that will take something that's in a vesicle and push it out of the cell. You can see in this diagram, first you have some sort of substance that's in a vesicle. Let me make that a little bit bigger. You have some sort of vesicle here, and then it fuses with the membrane, and its contents spill out of the cell, so out. And what this allows the cell to do is secrete membrane and secrete protein. So sometimes when that vesicle attaches to when it fuses with the cell membrane, then it allows the cell membrane to grow. And oftentimes, like as you can see in this, there's some sort of protein that needs to be secreted out into the environment, and that will also happen through exocytosis. Like I said, this is just vesicles spilling their contents out of the cell. The next type that we're going to talk about is endocytosis. Now, again, endo is into, and this is when you're taking materials into the cell. The two main types that we're going to talk about is phagocytosis right here and pinocytosis here. Phagocytosis, phago means food or eating, so phagocytosis is what we call cellular eating, and it's when the cell takes in large pieces of food. You can kind of see that here, where there's like a membrane forming around that food as it's going in. And pinocytosis is here. I can say that in a second. Essentially, phagocytosis can happen through two different ways. One, the membrane will just kind of bend inward, like you can see in this diagram, and then a vesicle will form around the food. And sometimes it'll actually form what we call pseudopodia, where it'll be more like this, you have this membrane that's like extending outward around pieces of food, rather than it just bending inward. But for purposes of this picture, we just have the bending inward. Now, like I said, pinocytosis is like cellular drinking, and what it does is it takes in these dissolved nutrients, like these little red dots you can see in this diagram, and it'll also take in some of the water in the environment. And kind of the difference between these two is really just what's being taken in, rather than the exact process. Also, some examples of phagocytosis. I should have mentioned this earlier, but if you know what a paramecium is, it's this type of protist that lives in like pond water and stuff. And this paramecium, when it eats, I'll draw a little diagram. We've got this paramecium, and when it eats, it'll see some food. Maybe it'll be like this little. See some food, and it'll be like this little red blob, and it'll you'll have paramecium. It'll like give these little extensions like this pseudopodia that I mentioned earlier, and so that's how it'll actually eat. In addition, your immune cells, actually the cells in your body that fight disease, there are certain types of them called macrophages and a couple other types that do the same process in order to eat bacteria and other pathogens and stuff. Also, there are certain digestive cells in your digestive system that do this in order to take in the food that you eat. So those are just a few different types of phagocytosis. Now, the last thing we're going to talk about is what's called receptor-mediated endocytosis. Now, this is a little bit more complicated, and it's got a little more of a mechanism behind it, but it's really not too bad. It's very similar to pinocytosis, but the difference is that a receptor, which is where it gets receptor in the name, binds specifically to a certain molecular compound. So it'll only be applying to one. So there's a certain molecule that's taken it in more concentration, in higher proportions. That's what it says here. This takes in one substance in higher proportions. So in this diagram over here, you can kind of see what just normal pinocytosis really looks like. So if there's two different solutes that we're going to look at, the green one and the blue one, and as the vesicle forms, it just takes things in the same concentration that they are outside of the cell. But what if the cell wants more of the red solute? What if it wants more of the red and less of the green? It can't really do that with pinocytosis because it's just going to go into the bubble. It's going to go into the vesicle just like it normally would. But with receptor-mediated cytosis, you have these little brown proteins that are going to bind to the red solute in order to take it in greater proportions. I've got a couple of diagrams to help you out. You can see right here you got the red solute, and it's going to fit perfectly into that receptor. And then as it goes, it's gonna stay in, whereas the green solute doesn't really fit in, so it'll just kind of keep going. When they start to fill, then you have a vesicle full of the red solute, and with only just a little bit of the green solute. And so this will allow the cell to take up a very high concentration of the red ones and a low concentration of the green ones. So that's really just how receptor-mediated endocytosis works. I hope this was helpful, and bulk transport is really just as simple as that.